So hello everybody. First of all, of course, let me thank Alveole, Hélène and Pierre-Olivier for the nice invitation to be part of this impressive webinar. Of course, we are not physicists, so we are very different from the other members, but it's also a very good opportunity for us to provide some light on a tiny microbe that is far from being well known, despite it is carried by about a third of the human population, and it puts the human population at risk for severe, for chronic, or for even life-threatening health issue, with a series of diseases that are grouped as toxoplasmosis. So you probably have guessed the name of the parasite, of the microbe, Toxoplasma gondii, which is a unicellular eukaryote which has chosen or evolved a parasitic lifestyle. It is a really strict parasite, so it depends on host organism to perpetuate its population. And it has been, I don't see very well for me. Okay, it has been then identified first in a rodent which is endemic from Africa and which is named the gondii, and it has been identified in 1908 by Charles Manceau and by Charles Nicole and Louis Manceau, sorry. And you can see here now, I'm blocked. Okay, the scanning electron micrograph of two parasites, you can notice, of course, the dimensions, so it's about several micron size, but also the typical bow shape. And bow means, or is said in Greek, toxon. So you now know the reason why we called it Toxoplasma gondii. In addition, I would like you to pay attention to the fact that at the both ends of the ellipsoid shape, you have two poles that are not looking similar. And in fact, it is a very highly polarized cell, and the polarity is maintained all along the life cycle. So we will go back to that because I think it's going to be important for the rest of the talk. So in addition to rodent and human being, Toxoplasma has an incredible wide spectrum of hosts. It can actually get into and colonize many, if not all, warm-blooded metazoan, including domestic, but also wild species. In addition, it is a hypercosmopolitan parasite. It has a hypercosmopolitan distribution. So the success of this parasite lies, at least in large part, in the fact that Toxoplasma is able to sense the change in the microenvironment and to respond timely by reformatting gene expression, so to drive the transition toward distant developmental stages through cell differentiation process. And for today, for the amount of time that we have, I will just restrict myself to two of these developmental stages called bradyzoite and tachyzote. Collectively, these two stages are enough for the perpetuation of Toxoplasma population, owing to the fact that they can reproduce by an asexual mode and also because they are transmitted from host to host. So, speaking about in this context of transmission, how do we get in contact with bradyzoite? In fact, all animals, including human beings, ingest bradyzoites with animal tissue that we are feeding on. So once we have ingested, because the bradyzoites are enclosed in a very protective envelope that we call a cyst, the cyst envelope is disrupted in the stomach mechanically and enzymatically. And this process releases the bradyzoites that are capable of then traveling in a very mysterious way through the gut to reach specifically the small intestine and specifically the enterocyte in the small intestine. There, it will invade the enterocyte and differentiate as tachyzoite, that's the first change between the two stages mentioned. And these tachyzoites are extremely active in terms of replication. So they will multiply and colonize the intestinal mucosa, all cells belonging to this mucosa. But your immune system, if it's competent, is very, very powerful or efficient at restricting the tachyzoid proliferation. Meanwhile, it does so, it also promotes the tachyzoid long-distance dissemination, at least for a tiny amount of the population that escape the immune killing. And in this tissue where they settle, they again retransform back into the bradyzoid that is a much more silent in terms of replication. So this parasite can stay cryptic as a bradyzoid for lifelong, unless you are subjected to immune balance disruption. So if you have a dysfunction of your immune system, then the bradyzoid that were staying quietly in this tissue will transform back into the active tachyzoid, leading to a massive proliferation of tachyzoids inside this tissue and creating tissue damage as well as inflammatory disease such as encephalitis, retinitis, and cardiomyopathy. That's just about the main disease of Toxoplasma, so toxoplasmosis. There is another way of getting transmission by tachyzoid is if the mother is primary infected with tachyzoid and the tachyzoid are not cleared mostly by an immune system because it's a primary infection, then the tachyzoid has the time to cross the placenta to infect the fetus and of course to destroy tissue. So our main interest in the lab was really to try to understand how do these tachyzoids locally disseminate in the tissue to find the cell in order to produce progeny. And this is of course a critical key step to the biology of the parasite, but also to the pathology associated with the parasite. So just to remind you, then when the parasite invades a cell, it will produce progeny, but then the progeny has to get out and actively egress from the host cell and then navigates in tissue, I would say in matrices from the tissue to search for new cells. And we really want to interrogate how they deal with this navigation step. So to do so, we just look at some parasites in 2D, just when they are on a substrate, like FCS-coated substrate, fibronectin, or even polylysine would be enough. And we can follow the trajectory by just looking at a marker that marks specifically the end of the base of the parasite, because we're using transgenic parasite expressing a chimeric version of this protein. And what we can notice immediately is that there is a diversity of cell movement, that there is intermittent activity, so there is up and go, but there is certainly a substrate dependency. The movement is unidirectional and it is very high speed, few micrometers per second. So it's a super high speed process. What is important to remember is that in 3D condition, we will find again the same the same gliding, the same mechanism of motion, but only one type, which is helical. And so we decided to focus more onto the mechanism underlying the helical gliding and to tackle down the force during this helical motion. So just let me remind you, the polarity of the parasite is a bona fide feature. So it is kept all along the life of the parasite. There is no time of polarization. The polarity is kept with an apical pole that directs the directionality of motion and is populated or featured by a specific appendage we call conoid, made of microtubule and actin together that forms this appendage, which is retractile or protruding depending on the time of motion and invasion. In addition, this apical pole is also populated by secretory vesicles that are in charge of delivering adenosine protein from the apical pole to, and which will be translocated backward during motion. And this is in concert with the with an actomyosin motor, you have different species of myosin involved that are taking in charge of the backward translocation of F-actin together with the adenosine that have been bound to extracellular matrix ligand and connected to the cytoskeleton. As a result, you can see that there is some material that is shed at the end of the parasite, at the basal end of the parasite, that is very characteristic of this parasite and that can be deposited as trail when the parasite is moving. So the model was very simple, but then it was kind of puzzling because when we put beads on the parasite, they indeed translocated from the front to the back, but they do so at a much higher speed than the motion itself, the cell motion itself. And they are quite nicely released at the back, similarly to what we can see with the material shed posteriorly. That would be the beads together with the material shed when the parasite is getting in motion. So we decided to tackle this puzzling issue by using live imaging combined with force microscopy, and that has led us to alveoli and micropatterning. So I just want to mention the members that who have been extremely important for this study. In particular, Yorgos Pavlo, who is now at the MIT doing his postdoctoral, but was really predominant, instrumental in driving these studies. And we did so in great collaboration with the expert in TFM and force microscopy at the LIFI, a neighbor institute of ours, with Delphine Debar and Martial Balland. That has led to a collaborative paper that has been published about a year ago now. So we decided to dissect the 2D helical gliding, and we realized that it was in fact a multi-step sequence. So we were able to track the back and the front at the same time, so to retrieve data about change in shape of the cell, while we were also able to track the speed profile and see that, you know, at each point, at each angular point of the trajectory, there is a pause. And this pause actually coincides with the deformation of the parasite. If we go back, I can stop, probably, okay. Okay, here, for example, you see this apical flexure of the parasite, and that coincides with the time the parasite is pausing. So the banding or the kink that we have named so far as a feature, as the early features of helical gliding sequence, correspond or coincide with the pausing time of the parasite. Then we ask, does the kink and pause could account for a specific adhesive event? And we did so by applying with Delphine Debar quantitative reflection interference contrast microscopy. So I won't go into the detail, but to make it short, we were able to retrieve the space, the local space where the parasite was indeed very, very close to the substrate, so close that we can imagine or hypothesize this is a true contact. And as you can see here, it's on the right, the segmentation due to, with the help of machine learning, really tells you that when there is a banding of the parasite, this apical flexure that we call a kink, coincide also with the apical contact to the substrate. On the green label that you can also see in the material of when looking at RECM, sorry, this is exactly the trail material I was referring to that also is in tight contact or with a high probability of contact with a substrate. So if we can define the sequence, we can say that when the parasite is resting, there is just a back adhesion and a flattening of the overall body of the parasite, but there is no apical adhesion. And contrast, when the tachyzoids start to be active and perform helical gliding, you can detect an apical contact early on, followed by the contact is spreading from the back, and an apical lift and twist to propel itself with an helical path. So the next step was to make sure that the apical contact at the kink could serve as a transmission anchor. And to do so, Yorgos introduced TFM with the help of Martial to measure the force during this helical motion. That was quite challenging because, as I told you, the cells goes very quickly, few micrometer per second, and also it's a very tiny force as well. So we had to work with a very elastic, a very soft gel in order to perceive the bead displacement according to the TFM technique or principle. And as you can see with Yorgos, we could see that during the kink step that you can see now, there is indeed a traction force that you can follow with the displacement of the bead labeled in the second panel. I can give it again to you. Okay. And you can see here the displacement that coincides with the traction force applied by the apical pole of the parasite, exactly coinciding with the kink, which itself coincides with the contact that we detect with QRICM. So TFM allows to characterizing the generation of the apical force that is applied at the kink, which triggers dragging of the body while the base rotated and perform the helical propelling. So then we wanted to really assess whether this force that we were uncovering was indeed important for driving or for controlling the ability of the parasite to perform helical gliding. And we searched for a technique to prevent the front adhesion without preventing back adhesion. And that's the reason why we went to micropatterning and try to use a composite micropatterning with alveole to assess the whole of this early contact. So to do so, we first define a completely, I would say, resistant or refractory substrate to helical gliding, and we find PLL was quite efficient. While PLL PEG was quite efficient, And so what we did is that we used a micropattern in which an area was precisely permissive because covered by a fibronectin layer, whereas the other part would be completely refractive resistant to helical motion, to gliding behavior, I would say. And we were interested, of course, at looking at what was going on at the interface between the two areas, looking at the behavior of the parasite. So we did so. And we were lucky to have enough parasites nice enough to approach this edge. And we could see that they perform perfect gliding on the permissive fibronectin-coated area. But when they approach on the base, the edge, they tried to go forward, but they could not. And then they twirl on their base, they rotate on their base and go back to the permissive area. So this one will show you maybe more in detail how the parasite really attempt to catch something on the substrate with the front pole, but because he can't, he decided to go back by rotating and gliding again in the opposite direction. That's exactly what we can see when we track the trajectory of the two parasites or one parasite shown here in the video. So, okay. Then to finish with that story, we tried to see whether or not the evidence we brought on a specific force that would be applied at the apical pole and that would be absolutely important in order to drive the helical motion. We just looked at whether or not we could have the same type of mechanism when the parasite is actually into a 3D microenvironment. And we use collagen fibers because it was an elastic support that could be very useful in order to see traction force operating from the parasite. So indeed, what Luis Vighetti in the lab is now performing the PhD and is continuing with Alveole to collaborate on micropatterning, has used rat collagen 1. We label the collagen 1 network with a bacterial derived protein that specifically bind to the triple helices of collagen, and we add parasite. As you can see here, the parasite can squeeze in between the fibers, but very interestingly, when they move, they really grab the fiber and rotate after the grabbing. So when we saw the kink, when the parasite grabbed the fiber, we could also look at the behavior of the fiber. And because the fiber is elastic, we could see that by looking at the different position at the very beginning and along the sequence of motion, you could see that the fiber is, so the first position is in blue, and the second is in green. I mean, the other are in green. So when the fiber doesn't move, it stays blue like that, cyan. And you can see that with time, the blue is distinguished from the green because the fibers has been pulled backward. And then when the parasite has gone, because it has propelled through the fibers, then you can see, due to the elastic property of the fiber, that the fiber comes back to the initial position and gives a cyan labeling. We can see that also in the last panel. It goes a little bit fast, but you can see the difference between the cyan and the green. So the next step that we now want to address is that can front and back contact point could be sufficient for accounting for helical gliding capacity of the parasite. And that is very important, but because as I told you at the beginning, it would really modify or revisit or shift the model of the membrane flow driving the motion of the parasite. And to do so, we used, the other question that we want to ask at the same time is that to try to combine the TFM and micropatterning to restrict the tachyzote on the base, but using different substrates, so different affinity for the parasite surface molecule to adhere to. And to respond to the first question, what we have designed is a micropatterning that could be either disc or line that would have 2 micrometer line that would be permissive to the parasite, would be fibronectin coated, but would be separated by a line that would not be permissive. In that case, it would be PLL PEG. And see whether or not the parasite can actually jump from one to the other, which would mean that it doesn't need to adhere on except on the anterior, on the apical and basal pole, like it was with the force that we saw or we uncover with the TFM force microscopy. So we are trying, of course, as a control, we will enlarge the non-permissive area, and we've tried to perform this with Primo. And at the very beginning, we were quite excited because we actually saw the gliding of a parasite coming from one to the other, and then, you know, but exactly on the line and then going from one to the other. But unfortunately, in the next assays, we could also see that some parasites were actually gliding in the middle, in the supposed PLL PEG area that should not be permissive to gliding. And even the kink that refers to this apical traction force was taking place in the middle between the two permissive lines that are coated with fibronectin and here labeled in red. And when we went closer to look at what was exactly in the line of fibronectin coated material, we could see that in fact, despite we tried to be very careful, we got some material in the middle that could be enough like stone in a river to allow the parasite to actually jump from one to the other and to grab this fibronectin in order to apply force. So we are kind of stuck at that time, sorry, and we try to improve, but we need to have a lot of high precision design because our lines cannot be more than 2 and 4 microns due to the size of the parasite that again is about 6 to 7 micrometer length. So with that, I will stop, and I will just mention that we also apply super resolution microscopy with Bastien Touquet in the middle of the photo, that helps us to to uncover or to shed some light on the twisting behavior of the microtubule that are already helical paths. We can see that they compress and relax like a spring in order to really propel or to drive the propelling of the parasite in an helical way. With this, I thank you all. Thank you.
Thank you very much, Isabelle, for this very refreshing talk, I would say. I really like this new, I would say, new, at least to me, cell models that we are not used to see. This is always very interesting to see. So I have a very, I would say, naive question regarding that. Is this helical gliding very specific to your Toxoplasma, or is it something that is very well observed in the parasites migration mode?
So in the parasite belonging to this phylum, which is a huge phylum that is called Apicomplexa and encompasses some very important parasites in terms of medical relevance, so, such as Plasmodium malaria causing the malaria disease. The helical gliding is something that we can see. Toxo is somehow the best so far known model of helical gliding, and it's always in the direction, I mean, in counterwise, due to the following the path of the microtubule cytoskeleton, of the cortical microtubule cytoskeleton. And when I showed you, for example, the bead experiment that we put on the apical part of the parasite and translocate backward, if you look carefully at the movie, they actually follow the microtubule trajectory. So it's not uncommon, but it's quite typical of Toxoplasma, and I think restricted to the phylum.
Okay, thank you. So we have a question from the chat from Philippe Carl, who is asking, what is the actual modulus of the soft gel used in your attraction force microscopy experiment?
Yeah,
and why do you use such low concentration?
Yes, it was extremely low because, in fact, the parasites, the term we use is gliding. So there is very, very low adhesion, and it's nothing to compare with, but that's also relating to the speed of the parasite that goes extremely fast, few microns per second. So to be able to detect motion, I mean, the displacement of a bead in the TFM gel, we really had to have something super elastic, and we had to finish with a gel of very, very soft gel. Yes, of course, it's a compromise between it's a trade-off between how to use, the stiffer substrate we use, the higher amount of parasite will be actually in the process of moving. By restricting ourselves to the setting where we could see bead displacement, we also have a challenge not to see a lot of parasites. So we managed to work on about 20 different parasites to measure the force, the overall force when the parasite moves. But of course, and it cannot do much more than 1 or 2 helical cycles it pauses in between. That's because of the parasite. It's a very tiny parasite. Huh? It's nothing to compare with a big mammalian cell growing on a substrate.
Mm-hmm. Okay, thank you very much, Isabelle, again for this presentation. We